And we have published more than 10 books, including Market-Driven Plant Breeding as our flagship publication with Springer. We have trained professionals not only from India, but across the world, including Italy, Spain, New Zealand, Sri Lanka, Bangladesh, Nepal, Afghanistan, Brazil, Nigeria, Kenya, Ghana, and you name. And we also have 12 CSR certification, naming us eligible for CSR projects. Now, I would like to take a privilege to introduce our moderator, Dr. K.K. Narayanan, Founder Director ATPBR. His career began as a scientist at SPIC Science Foundation, later leading crop transformation at Monsanto Research Center, Bangalore. In 2001, he founded Metahelix Life Sciences, and currently he is the founder managing director of Styka Seeds Private Limited. He actively serves as a scientific task force under DBT, Government of India, and Karnataka Vision Group of Biotechnology, driving innovation in agriculture and biotechnology. It's a privilege and honor to have you with us, sir. Now I request Narayan, sir, to please take the floor. Narayan, sir, did you join the session? Yeah.
Yeah, I am here.
Please, sir, take the floor.
Can you hear me?
Yes, sir, I can.
Can you hear me? Is my voice clear?
Yes, very clear, sir. Very clear. Please go
Hello.
ahead.
Yes, sir. Good morning, sir.
Is my voice clear?
Yes,
Sir,
yes.
your voice is
Good
very
morning.
clear.
Are you able to hear me?
Yes, sir.
You are loud and clear, sir. You are loud and clear.
Aparna, are you able to hear me?
Sir, we can hear you very well. Call him up.
You are
Okay, so good morning to everybody. So good morning to everybody.
Good
Those
morning.
who are joining from other time zones, my greetings from Bangalore. So the topic for today is about BT cotton. And you know, BT cotton was a watershed moment in the history of agricultural technologies of this country. You know, the cotton productivity and production increased in the initial 7 to 8 years. And I think over the last decade or so, maybe from 2010 to 2020, there's been some kind of a technology fatigue. Resistances have been developing in pink bollworm especially, and new technologies were not coming, or replacement technologies were kind of somehow hindered from reaching the market. Can I call it some kind of a technology constipation? That wouldn't be entirely inappropriate during that period. But I believe now, after the COVID, particularly, there's been some stirring happening in the technology, agriculture technology sector, particularly in terms of testing some of the crops which have been almost shelved, which have been taken out. And who is a better person other than our speaker for today, Mr. Balasubramani? I know he started his career in my alma mater as an assistant professor in Tamil Nadu Agriculture University, but then soon moved to the Central Institute of Cotton Research. So when the BT cotton was in the initial days of making an impact on Indian agriculture, I would say that he had a ringside view of the happenings, and I'm sure he has been following it. And I cannot wait to hear about the updates and what is happening now, what we can expect in future. So Balasubramaniam, the stage is yours. Please take us forward on the story of BT cotton. Thank you very much.
Thank you, sir. Thank you so much. Yes.
I would suggest that we have about 1 hour of time.
Yeah.
If you can make your presentation in about 40 minutes or so,
Yeah,
that
yeah.
will leave about 15-20 minutes for discussion. So we
Yes,
can have a participatory meeting. Will that be okay, Mr. Bala?
Yes, sir. Yes, sir. Fine, sir.
Okay, let's do it that
Yeah.
way. Bye.
Shall I start?
Please go ahead.
Yeah.
Yes, please.
So can you see the slides?
Yes, very clear. Please go ahead.
Yeah. First of all, thank you, Madam Aparna, Madam, for inviting me to give this lecture. And thanks to the Narayanan sir, we had a great association during the biosafety experiment and all, especially NDRA Karnal. We all went to conduct these experiments. So after a long time, I am also seeing in this webinar. Thanks sir, for inviting me to give this lecture. And Rahul also thanks for arranging all this webinar. So now I'd like to start the topic of current status of transgenic cotton in India. The slides are moving.
Yes,
Yes, sir. Yes, sir.
So this slide is just to give an introduction about this cotton. The major cotton growing countries, you all know that India, China, US, and Brazil. So of course, India is the more than 12 million hectares are being cultivated by 4 million small and marginal farmers. You can see that interestingly, around 36% of the world cotton area comes from the India only. However, the crop production, if you see that the China leads around 30 million bales in the current season, followed by the India 25 million bales. So here I listed top 10 countries growing the cotton. Among them, that 4 countries are major contributor: China, India, Brazil, and US. If you look at the 23 to 25 million, the year 23 to 25, so I had given that the list, the cotton productions. China, of course, it's slightly reduced in the current season, whereas India is constantly increasing. Brazil and the United States are constant in the production of cotton. Look at the cotton acreage and yield in the last 5 years. So India, in these 2 years, if you see that there is a area is increased, but proportionately the yield is not much significantly increased but compared to the previous 2 years. So this is the last 5 years status of the cotton acreage and yield of India. So regarding the production consumption of cotton, you can see that India is the only country where all the 4 species of cotton can be grown. And of course, the hybrid technology is available in GE Sutam, and BD technology is available in the GE Sutam. So the hybrid technology is successful in India. And India's major cotton growing zones are here listed here. Of course, the Odisha has joined in the one more states. Now you can see that interestingly 13.6 million lakh tons of cotton is coming from the Odisha. Regarding consumption of the cotton in India, you can see that we are the second place in the world, the estimated production of 323.1 lakh bales. But of course, that almost 90% of the cottons are utilized for the Indian, our country only, consumptions. But only the export and import will be very less with compared to the consumptions. Of course, the export quantity is reduced drastically. Now this year it's estimated 18 lakh bales of cotton. But in case of import, mostly the Egyptian cottons are imported. Even when we visited some of the textile in Nagpur, Maharashtra also, they told that mostly we are importing the Egyptian cotton one. So that way the Egyptian cotton is getting import quality. So coming to the challenges for the cotton research, really the cotton is a challenging crop because it has the many challenges, especially the environmental and biological challenges. If you take that, pestilence disease is the major one. So more than 163 pests are attacking cotton, but majorly around 15 insect pests are important. So that is making a loss of nearly 30 to 60% loss every year. The second issue is the weed competition. It is going to play a major role in the big challenge for the cotton farmers. Of course, the third one is the climate change. So there is a sporadic and untimely rainfall also leads to the big challenges for the cotton farmers. But that is not in our hands. Regarding the regulatory and policy challenges, there is a big challenges. When we started the transgenic cotton work and biosafety work and all, there will be limited hurdles or regulatory issues was there. so that we could complete it. Even that takes nearly 3 to 4 years to complete the biosafety studies. But now, since the biosafety is dynamic issues, so you keep on adding more hurdles to release any new events. It takes nearly more than 5 years to comply all the biosafety issues. And also the policy implementation also another important things. For example, if you take the RIB or the ST cotton, so these all the policy issues. It's taking long time to resolve these issues. These are all the big challenges for the cotton research and cotton farmers. Coming to the research and development challenges, we have limited germplasm limitation. There is a exchange of germplasm within the country or the overseas also. It's a big challenge for the cotton. So fiber quality testing is very important because most of the new varieties or hybrids are failed because of mainly the fiber quality. So if it is not the benchmark, it's not qualifying, then most of the hybrids or varieties are failing to qualify and release. And also we have limited R&D facilities. Nowadays the cost of things are increasing, so we have the limited R&D facilities. And agriculture infrastructure and constraints are there mainly for the genetic purity and inputs. And there is, of course, the last one is the economic and marketing challenges. There is unpredictable prices and returns. These are also big challenges for the cotton farmers as well as the cotton research. However, we are making a lot of efforts to address these challenges thanks to all the cotton breeders, allied subject and biotechnologies to solve all these perceived problems. But the interesting part is that we are all fighting with the insect pest. Mainly, I put that one slide that is a tug of war is going between the insect and the scientists, because sometimes we are winning, many times the insects are winning. It's overcoming all our results. So this is the tug of war only. Although we are making a lot of efforts to develop the bollworm resistant cotton, we are making a lot of technology, especially precision agriculture technologies, climate smart cultivation methods, and genome. Recently now we are going for the genome editing techniques, especially with the CRISPR-Cas system. What is the ultimate goal is that we'll have to enhance the cotton yield and the quality cotton and resilience to the environmental stresses. These are all our main objectives where we are addressing all these challenges. This multidisciplinary approach only we can help to overcome all these issues. Interestingly, we are the pioneer in the cotton hybrid technology. The first cotton hybrid was developed in Gujarat. C.T. Patel has developed. He is the father of the hybrid technology. Because that particular technology helped this successful commercialization of the private companies, the BT cotton technology. Coming to quickly the BT cotton in India. So the first BT cotton, when we joined in the service, I joined the service in 1993. That time there was no any BT cotton work or gene construct, nothing. Just only tissue culture we started. So, but that time there was some report that Parlak 1990. So he's inserted Trivonase gene in cotton Quokka 312 using the agrobacterium incubation. This was the first report. So from that time onwards, that it is developed, it's got the attention in all over the world. That first in 1996, the Monsanto commercialized the first BT cotton in the US. In India, I think Myco has started that work in 1995, got the seeds, then they undergone all this field trial, the ICR trials. Finally, they got approval in the May 9, 2002. So this was the first BT cotton genetically modified commercially available in India. So this is some of the timelines I had given here. 2002, the first Transgenic hybrids were released, 3 hybrids. And 2004, RCH2. So RCH2 become famous in the quick period because this was the genetic background was excellent. So it taken the lead in the cotton, BT cotton. Then 2005, almost northern states also it's allowed for the wider cultivation of the BT cotton. 2006, the Bolgot 2 was introduced, where these 2 genes were introduced to break any resistant development by the bollworms. So that was the concept. From 2002 to 2014, you can see that GSS released more than 1,167 BT cotton hybrids. But that time, no varieties were there. All were hybrid cotton only. So then there was some discussion that there are so many hybrids that keep on releasing by GSE. So there should be some restrictions. So they stopped after a particular period that, so we cannot release more any events. Of course, there was not much events. Only 5 or 6 events were released. After that, there is no any new events were released. So they transfer this the responsibility to the ICR. So of course, this is the 6 events. Of course, the 5 events are available in India. So the Monsanto, that is one. The first one is the Cry one is Bolgard 1. The second one is Bolgard 2 with 2 genes. North seeds with the fusion genes, and JK seeds with the Cry 1 AC, and Metallics KK Narayanan Sarka. So that was the 2008. It was released in the event name is 9124 Cry 1 AC, Cry 1C. So our event was simultaneously released with the Metallics only, but there was some problem with contaminations. So this was identified. This was developed at Dharwad, but released from the CACR Nagpur. So due to this contamination, this was ICR was stopped, discontinued this event. So primarily now the 5 events were released. It's successfully it's going on. However, this now these 2 genes only, most of the hybrids are released, varieties are released nowadays. Now the responsibility is given from GSE to ICR. So the GSE is entrusted the responsibility to ICR in 2017, especially CACR, to evaluate and release the new BT cotton hybrids and varieties developed by the public and private companies using the notified deregulated events. So ICR and CACR has taken that this technology to through ACRIP mode, assigned to evaluate the BT hybrids and varieties through the ACRIP centers in all the cotton growing zones, especially in the Kharif seasons. So then ACRIP has generated the comprehensive data on standard parameters. So they normally ACRIP is using some of the standard operating protocol. That is was they applied here also to release the new hybrids and varieties. If you see that these are the major standard procedure protocols, any new varieties or hybrids has to qualify. Unless qualify these criteria, then it may not be released. So mostly the trials were conducted 3 years. So now it has come to 2 years also, some of the entries. So mainly what we are looking for is the yield performance. It should be better than the local or national check. Then the fiber quality, it's most of the entries are qualifying, but some entries are, if it's not qualifying, means mainly this yield and the fiber only. It's major issues. It's failing means. Then another important is the BT Cry protein expression. So it has a big story. When this was transferred to the ICR, so we were testing the protein. So in our lab only we were testing the BT referral lab. So what happens, as per the ACRIP protocol, we used to take the average of the Cry protein expression, all the tissues as well as the different intervals. So because of that, the guidelines of the passing criteria, Cry 1 AC should be 4 ppm. 4 ppm it should be there with the dried weight or fresh weight, or the Cry 2 AB should be 280 ppm. So this was the criteria as fixed by the Gazette notification the Ministry of Agriculture has released. So, but if you take the average of the Cry protein, it is not qualifying. No entry was qualifying. So this was going on in nearly 2 years. We used to continuously meet with all the firms, especially Myco, Nuzivid, Rossi Seeds, all these major players. We used to regularly in the Delhi and discuss how to solve these issues. Finally, our former Director General, Dr. Mahapatra, he called one day a meeting of all the people. Then he asked us, what is the issue? He told that, sir, the Cry protein is not, no entries qualifying. So what shall we do? So he saw that Gazette notification. Then he told that why you are taking average, because there is no any mention of the average of all the season, all the variety, all the tissues. Then why you are doing? Then if you do that, nothing won't get qualified. So that way you resolve that problem after seeing the Gazette notification, because it's clearly written that So the 4 ppm of Cry1AC and the 280 ppm of Cry2AB should be express any tissues at any stages. So that way we solve that, resolve that issues. So since then we tested. So mostly 60 days of or 90 days of the tissues, especially leaf tissues, mostly square also, it is coming in the 4 ppm. So it's almost invariably all the varieties or hybrids were qualifying with, if you take any tissues at 90 days. 90 days, mostly 90 days, you can see the peak of the tri protein expression. That way, this problem was solved by our farmer DG. Then the other aspects of agronomic and pest resistant traits and endocrine biosafety compliance, all those things we'll have to monitor. Then only this will be released, the new varieties. So ICR is released BT cotton hybrids and varieties. You can see that 2017 to 19, there is some recognized because of this protein expression things. It was not released. In 2020, the first release, the nearly around 28 to 30 BT hybrids and varieties were released. So you can see that since 2020, so you can 163 BT cotton hybrids were released and 3 non-BT varieties were released. So here I've listed here some of the hybrids released 2020 to 2023. Here I put that some of the varieties also. Here I have given listed that the release by the ICR. Here you can see that till December 2024, 163 BT cotton hybrids were released. But last year, 22-23, alone 45 to 46 BT cotton varieties and hybrids were released. But 24-25 is pending now. It is under the considerations. Almost any time, I think after the meeting of CVPRC, then they will be declaring that the 24-25 new list of hybrids and varieties. So this is the CACR released BT varieties. Of course, this was the deregulated events of Cry1AC. Total 15 BT varieties were released so far by CACR. We all were this part of this program. So now we'll see the impact of BT cotton. So if you see when it was introduced, the cotton farmers were very enthusiastic, and almost the adoption rate was more than 90%. All the Indian farmers were this adopted the technology. It has the mixed kind of results. Initially, if you want to divide this, the increased and decreased stage, like the Narayanan sir initially explained that how that initially it's shown that benefit, later what happened. So this I can divide into two parts, from 2002 to 2015 and 2015 to till now 25. So we can compare all these the parameters. So first if we see that yield, initially it was increased. Now it is kind of stagnation only. There's so much increase. Second, the farmer's profit, if you take that, initially it showed that increase because of the less input cost, especially the spraying. But now it is increasing the cost of, the decline the profit because of this input cost is more. Pesticide usage is initially decreased. Now it's increasing due to the resistant development by the pink bollworm as well as the sucking pest we are using it. Input cost increased. Of course, this is remaining high only. The farmer adoption is increasing rapidly. Initially, it's increased. Now also it's keep on increasing, but not much. So now we see that GM cotton events delivered the obvious benefit to the Indian farmers. But only thing is the technology, if you want to make sustainable, so we'll have to support the strong agronomy practices, seed systems, and ongoing pest management, resistant management for sustainable impact of the new any technology, especially the BT cotton. The major issues now we are facing is the pink bollworm. This is the major impact the cotton farmers are facing now. So what are the problems how this is developed? Now we'll see that. Till 2009, there won't be any problem. It was under the control, all the bollworm complex. But in 2009 to 10, so we found that CSR only observed that there is a pink bollworm incident in some of the, especially Gujarat and Maharashtra. The incident was found. Then we listed out what are the problems, why this was developed, this pink bollworm. So one is the cultivation of long duration crop hybrids. So that provides a continuous availability of the flowering and fruiting stages. That's why it was coming. In fact, there was a notion that pink bollworm comes in the late stage of crops, but it's not coming. Now it's coming in the early stage also, because it is getting continuous cropping seasons, continuous materials for the flowering, fruiting bodies for the continuous generations. And the early and the extended cotton cropping, the non-compliance of refugia, that is a major problem of, because it's very difficult to eradicate completely any species of the insects. So that's why the refugia concept was there, but farmers failed to, or they may not be knowing that the concept of the refugia concept. So many people, they were throwing that non-BT seeds. They didn't practice properly the refugia concept. So that is also one of the reasons. Then storage of contaminated cotton, also another reasons where this continuously is getting the pink bollworm incidents. Another interesting is that pink bollworm is the monophagous pest. It's completely dependent on the cotton plants, because in fact this pink bollworm is not the leaf feeder. It prepares only flowers and bolls only. Because of that, it is it's survival of the fittest. It has developed this resistant to overcome this BT protein. Now there is no any technology available except the management practices. So even now, pheromone trap also one of the IPM technology we are introducing in our management practices. So now we have developed the AI model of pheromone trap to control the pink bollworm and other sucking pests. So the best approach, what the Tabas, Nick, they mentioned that, so better to go for the BT toxin only to overexpress it or produce more protein in the bolls. So that only can help, he said. However, what we observed is the highest protein is expression you can find only in the leaf, followed by the squares and bolls. Mostly the boll end is coming very less protein. This is also reported by the Nagappa and Kadi. So because of that, the insect is developing the resistance. So this is also one of the reasons why the insect develop. So now we'll see that how that refugia in bag concept has brought to the Indian scenario to overcome this resistant development. This you can see the notification is given in 2016 only. That time it was separate bag, it was given 120 grams. But later this in the RIB concept, they instructed that 90% of the seed should be BT and 5 to 10% should be non-BT accordingly. So this notification is given. So this is the RIB guidelines. BT should be 450 gram, means it should be mixed with 25 gram of non-BT. So that is 475 gram. That means the BT seed purity should be 90 to 98%. Non-BT should be 5 to 10%. But if you see that this is very narrow range, so then it has to be monitored also. So ministry has asked us to standardize the working sample size of RIB complaints to monitor the RIB complaints. Accordingly, we have taken the project. So then we did this experiments. Statistically, we proved that so working sample size should be 180 seeds. Because if you take the 90 seeds, so the LSF plate 96 plates, because we need this blank control, positive control, negative control. So it goes 6 well for this standard. So we need the 90 seeds only will get 1 plate. So 90 seeds we can do that one means. So we are taking the randomly, because whenever the seed inspector collect the seed sampling, what he do is he takes the 3 parts of the seed. 1 part he keeps with him, 1 part is sending to the referral lab, 1 part is to the owner. So again, this 1 part is coming to the beta referral lab. We are taking these particular portions. So we are making into 4 parts, and each 40 seeds we are collecting. So if you collect and see that, so what happens? we are selecting. If randomly we select that, if more than 3 or 4 seats are not coming in the selections, so what happens? It will fail. So because the range is very less, 5 to 10%. So that's why that 90 seats we are testing. It's most of the entries were failing. So then statistically proven that 180 seats, if we take that, almost all the entries, if they are passing, the criteria is pass that one. So that way we recommended that 180 seats are must to do that each flight. So then what happens? There is some practical issues that we need 2 plates of ELISA for Cry1 AC and 2 plates for the Cry2AB. So 4 ELISA plates are required. So to monitor this RIV complaints. Then when we recommended to the ministry, then there is again kind of a discussion that because there is issues. Mainly what happens, the state seat testing laboratories, they told that it's very difficult to make purchase 4 plates and do for one sample because they are getting hundreds, thousands of samples. It's very difficult to purchase. It's a costly affair, they said. So because of that, still that we are awaiting for the gazette notification for monitoring the RIB complaints. One more question is coming that whether really this RIB complaints is going to solve the pink bollworm resistance. So this is again big challenges for the cotton farmers as well as the companies. Now I listed out already. I told that what are the key challenges for the BT cotton farmers. So I told that that's pest resistant and secondary outbreak of pink bollworm resistant. Then secondary pest outbreak. So also this all some of the we'll have to go for more spraying to control these incidents, and also yield stagnation. These are some of the important issues. Profitability concern and increased cost of production. Then environmental and agronomic issues like non-compliance with refugee policies, water demands and climate vulnerability, or legal unapproved varieties. These are all some of the issues where this technology is in the big threat. So any technology, if we take that, so we should have this, the diversified pest management or improved policy enforcement and more sustainable agronomic practices to secure these long-term benefits. So coming to the now new trials, because whatever the denotified events only, we are ICR is releasing new BTR varieties or hybrids. But in case of new events, again GS is only conducting all these biosafety trials, then biosafety research trials, then they are releasing it. Now these are some of the I listed out new events are under the trial now. As of 2020-25, several transgenic cotton events under the BRL trials in India, mainly to control the pink bollworms. So the GSC, so what now they told that you'll have to take no objection certificate from the state government. So then again the issue comes that the state government is, some states are giving, some states are not giving. There are also some of the challenges for all the new events trials. So here I listed out all the transgenic events in BRL trials, especially this is for the pink bollworms. I could go through the literature, all those things they mentioned that it's mainly for the pink bollworm management. First we'll see that bioseed research India. So the event name is the Biocot X24A1. Initially it was the Cry2AI. Now they changed to Cry8EA1. So this is the gene. Now they are in the first year. I think they complete the first year. The trials are approved sites in Madhya Pradesh, Karnataka, Andhra Pradesh, Haryana, Punjab, and Gujarat. So they were the trial is going on. Then second one is the Anku Seeds Private Limited. They have the event is NBRI PGK519. This MCry1EC. This is the first year BR trial they are going. And also they have the 2 breeding stock hybrids containing the event expressing these stacked genes, the Cry1EC, Cry1AC, and Cry2AB and NPT marketing. So this is the 2 stacked hybrids they are testing in the BRL1 trial first year. Coming to the Rasi seeds, they have the synthetic Cry1 chimeric genes for the pink bollworm management. So this one Cry1C. So this is the first year trial is they applied. Of course, their NOC from the state government is under the process. Ajit seeds, they have the event selection trials. This is the 5 GM cotton event lines they use. Has got 1012, 105. The gene is the Cry2AA. This is pending progression in the BRL1 phase. GSA allowed for the event selection trial. I think they can take in the controlled conditions, confined conditions, controlled conditions they can take. Then the Rallis India Limited, they have this is the events, and this BRL1 and BRL2 trials, these stacked events, is approved for the stacked cotton events resistant to the pest, especially the Helicobacter pyrgera, Spoda prelatora, and herbicide tolerant. This is a combination of the 3 genes. So all these events are as of 2025 Kharif season, with some events are entering into the second year, and most of the other things are the beginning only first year. However, the final commercial release it will be after completion of all the biosafety experiments, especially the lab animals and the large animals, all the allergen tests, all those things they'll have to undergo. Then only they will be released by the GS. Coming to the ST cotton, this is again the major issues, and it is some of the policy issues also is pending with the government of India. Here you can see that current status of the ST BT cotton in India. So ST cotton, that is herbicide tolerant cotton, resistant to glyphosate herbicide. So an expert committee, of course, before that I want to tell that one, the MICO already tested in 2009 to 11. Then the report also given, but due to some reasons, this was stopped. Again, they've given permission in 2022 to 24. They gave the 3 years data. We had also conducted experiments, and we are given their recommendation, positive recommendation. However, this is the proposal is still pending in the with the GSC. But before that, 2017 onwards, there is a big issues that the newspaper, daily newspaper, we are used to get that one. ST cotton is grown illegally in many states, especially Gujarat, Madhya Pradesh, Maharashtra and Telangana. So 3 states only major it was there. So it was not released officially, but people are cultivating in like Gujarat, Maharashtra, Andhra Pradesh and Telangana. So you can see that the illegal BT seeds have created large parallel market valued around 550 to 600 crores. Only thing is concerned here is the farmers are, of course, good technology, farmers are ready to adopt it. But the farmers are cheated by some of the people. That is the big issues because of this illegal cotton. So we want to curtail completely the illegal cotton. There is DBT also sent to one committee that we were also one of the member in that committee to monitor. We collected the samples, we brought that, we tested that, we found that there are a number of, because randomly we selected the samples, especially the roadside only. Even there we could find that there is ST cotton contamination. Contamination or many intentionally they were growing in the BT cotton, the ST cotton. So what are the challenges found look for the ST cotton is, government is planning to legalize the ST BT cotton, especially to curtail this declining of the cotton yield. But the regulatory discussion is going on. But before we coming, so that BT cotton is illegally they are grown. Only some kind of reservations stopping that one legalization of ST cotton is. So many people, many NGOs were giving that concern about this ST cotton. So their main concern is that it will be indiscriminate glyphosate usage. This will be the problem for the environmental and health issues. Secondly, there will be chances of super weeds formation. That is there, of course. Some of the weeds already 100% weeds control is not there. 1 or 2 weeds it's there. So that there will be some super weed formation also. So that is another concern. Risk of gene flow will be there with wild relatives. Okay, that is loss of biodiversity. That is another issue. These are all issues listed out by some of the NGOs. Then reduction of biodiversity, especially the edible weeds. Here you listed some of the edible weeds by the human. So this also will be affected, and contamination of the soil, water, and soil and water, and loss of forage for the cattle feed, and herbicide residues in feed. These are some of the negative impact of the ST cotton they listed out. But positive, you say that nowadays the farmers facing the problem of labor, and also the tillage maintenance will be good. So that way the farmers want this technology, because it's not really so people are going for the illegal cultivations. Okay. So in India, there first initially the Roundup Ready was introduced, tested, of course. Introduced means they tested in the experiments. They found that it's not effective. Then even this MON 88913, so this Roundup Ready Flex cotton was tested. Now till this event only tested. But here we are getting the samples, unapproved ST cotton. Especially these samples we tested in our BTRF lab. So I listed here. So till 2017 to 2022, we have tested so many samples. We found that these ST samples, these are some of the percentage of the this is the seeds were supplied by the seed inspectors. Then we conducted the hands-on training to the most of the seed testing laboratory officials, especially the officials from Gujarat, Maharashtra, Punjab, Rajasthan, and Narayanan. So then since 2020-21 onwards, STL labs were testing their respective samples. So only the code samples we were testing. Now this also we stopped that. Only the recognized laboratory can test. There are some 4 recognized laboratory by the Ministry of Agriculture. So their lab can be tested the code samples. Now we are sending back to all the ST samples referred by the code. So here I would like to tell about this another issues of the cotton is the CLC with transgenic cotton also. What are the transgenic cotton status? I would like to tell here. So it has spread extensively in the northwestern India. Around 1.2 to 1.5 million hectares are affected. This is a major 90% of the regions where up to 90% were infected by these northern regions, especially Rajasthan, and Rajasthan only major. So the reason would be the alternative hosts were available for whiteflies. Especially the whiteflies is the main cause for the spreading the CLCV disease. So if you avoid this one, brinjal, okra, tomato, tobacco, and sunflower, then we can minimize this. This again management practices. These are 2 weeds like abutilin and solanum. This you also can avoid for this, because this give the host for the whiteflies. Of course, there is no any transgenic technology available for leaf curl virus. Although we tried with this antisense RNA technology and RNAi technology, but it was not successful because this virus keep on evolving. So it's very difficult. It's a big challenges for the transgenic biotechnologies, because till now there is no any transgenic cottons are available. Although the Pakistan has developed some of the transgenic events, but they told that it is the viruses keep on evolving. So it's very difficult to develop any transgenic technology. Probably with the help of the CRISPR-Cas or multiplex CRISPR-Cas only can help to curtail this incident of the leaf curl virus. Still then we'll have to go for the management practices. Coming to the STPS systems. So initially this ICR, CACR only, Nagpur only initiated this technique. Although it is an old technique, but Dr. K.R. Kranti, that time he was the director. So he initiated that and told that why don't we go for the high density production system, because most of the countries are using this high density. Only India is using the hybrids. Because of that, yield is reducing. So he initiated that work through the SDPS. We can increase our cotton productions. So that way it started in 2010, but later he left that organization. Then later Dr. YG Prasad joined. So he told that instead of going with the non-BT with SDPS, it won't work. So we can go with this SDPS with BT cotton as well as hybrids. So many MOE were signed with private concern to go for adapting their hybrids to under the SDPS, and that way it's Maharashtra and Telangana. We were successful and started this testing of the SDPS system. And of course, Dr. Venugopalan's agronomist, he was the main player of agronomists to take this into the many NGOs, many companies. He was the coordinating these techniques. And thanks to all the three people, Dr. Kranti, Dr. YG Prasad, and Dr. Venugopalan. So this high-density planting system, mainly what happens, we need the compact and short duration hybrids or varieties to fix the closer planting space. So main purpose is to increase the population, plant population, especially 26,000 to 29,000, because initially the traditional method was 7,000 to 8,000. So this way help to higher the yield. So then under the Ministry of Agriculture and Farmers Welfare and also Ministry of Textile supported this program under the NFSM project, and it started 21 to 22 and it's continuing also. It's Maharashtra, Telangana, Andhra Pradesh, and they lead now SDPS in adaptation with 22,000 acres under the Maharashtra SDPS projects. And also the government is supporting incentives 16,000 per hectares, and many people were participating now. So ICR and giving ICR and ICACR has signed MOU with the private sectors, and we are promoting this SDPS system. Hope that this will deliver more yield and the day and especially increase the average yield of cotton in India. So the spacing here, plant spacing mainly the 90 centimeter to 15 to 30 centimeter. So then this can be used in the shallow or medium soil using BT cotton hybrids and pest resistant. Input outputs. The main thing is precise canopy management and use of growth regulators is very important to suit the mechanical harvesting recommended. And also this the challenges for the future direction is that SDPS success depends mainly to tailored pest management and farmers training, because farmers are what happens. Some farmers, although we told many times that we should not go for a thinning, so some farmers went for the thinning. So farmers trainings are required to understand the SDPS system in India. And also we'll have to develop the more compact and pest tolerant early maturing varieties to scale up this SDPS system. Then only we can expect to lead the cotton productions. So coming to this organ, just I put that one or two slides to tell about that why this this is not coming under the Transgenic program, but this is very important to know that because there is a keep on increasing the demand of the organic cottons, organic cotton demand. Even many NGOs were approaching us to give for the organic non-BT cotton to suit the organic cotton. But we had given, of course, some of the varieties we had given, but their demand is more of high yielding type of non-BT cotton suitable for the organic cottons. So that way, these two varieties were released, this VJ SGF1 and this is the arboreum varieties, and also this one is the Hirsutum varieties. So these varieties they released in the our government of India. So this is the two organic cotton is released. What is the key features and benefits of the organic cotton? We need the participating breeding program involving farmers and scientists, reflecting the farmers' knowledge and the needs. But also we'll have to need the focus on genetic purity and biodiversity, avoiding the GM contamination. Mainly the organic cotton, where the problem is that the 90% of the area comes under, or more than 90% of the area comes under the BT cotton. So it's very difficult to monitor and maintain the genetic purity of the organic cotton. So if you, even now also we are testing our field conditions, there is a 5 to 10% of the cross cotton, because the cotton is often cross-pollinated crop. So you can find that there is a contamination of the BT cottons. So that way it's big challenges for the producing the organic cotton. Even if the one cotton seed also found that positive, then the entire lot will be rejected in the organic certifications. And also because a lot of demand is coming, the seed availability, it's very, very big challenges. And also fiber quality, whatever we are the organic varieties we released, all those things, the fiber quality is very poor. And also yield stability also is very important to maintain the sustainability of the organic cotton. and support the small farmers' livelihood because it would strengthen the organic cotton value chain. And also we need to incorporate modern sustainable farming practices so that this organic farming will be successful. So finally, I bring one or two slides for the branding of Indian cotton. So this is the government of India is initiated to brand that Indian cotton. Mainly the objective is to branding the traceability and certification to promote the Indian cotton to domestic as well as the overseas market. Because nowadays everybody is telling about this branding and traceability certificate is very important. That way, the CCI, that is the on behalf of the government of India, CCI is signed with the that is Cotton Textile Export Promotion Council, popularly known as the Texprocil. So they now giving the fund to trade and industry bodies to brand this Kasturi cottons. So already they have given this money. Then also they given the target period. So to expedite that, what would be the outcome of this project branding program is that we can reduce the import dependency. We can improve cotton quality, boosting yarn, fabric, and garment exports. It's a better price for the farmers through the adoption of the best practices. Because within our QR code, now we were able to trace this cotton, Indian cotton, we can trace it. So this way, the Kasturi cotton market is now we are under we are taking that lead. So the certification protocol is finalized, mainly to standardize the best fiber quality, like 29 mm to 30 mm long staple cotton is standardized. Mainly the enable accreditation testing is now we have finalized. The scale of production participation, you can see that 379 supply chain members on board. Already they have joined to this Kasturi Cotton Bharat. So here we can go for the traceability technology and digital support and global launch, so that the last the plenary meetings in ICSC Mumbai, they have taken that the global launch. So the Kasturi Cotton My Stamp unveiled by the Union Ministry of Textiles. So this is about the branding of our Indian cotton. Regarding the other GM crops in India, so fortunately only cotton is approved for the commercial cultivation in India, but other two crop like the BT brinjal as well as the GM mustard, these two were approved for field trials, and also they cleared the environmental clearance, but due to some reasons this was not commercially released. But in otherwise, there you can see that more than 20 crops are under the various stages of the research and field trials. Because major food crops, especially you can see that rice, wheat, maize, brinjal, potato, sorghum, mustard, groundnut, cauliflower, okra, chickpea, pigeon pea, castor, and sugarcane. So many crops are under the genetic modification, but only thing is that is it really required? Because government of India has the kind of reservation that the food crops there is a restriction out of restriction, because mainly they are worried about that long-term usage of the foreign genes. What would be the effect? That's why the biosafety research has to be strengthened and monitored, because this very technology should be safe to the human as well as the environment. So that way we are making all these safety measures to release into the environment.
Are you done, Bala?
This is last slide, sir. So if you look at the global agriculture, you can compare with China. We are very blessed with that. We have the best cultivable land area is more in India. Because of that, we are able to feed multiplying more. Because now more than 145 crore people, we are able to feed them, because mainly we are blessed with all cultivable lands. So my conclusion is that with the shrinking resources and exporting populations, deploying cutting-edge technology should be encouraged. Nevertheless, caution should be taken, especially the biosafety and environmental environment, to apply such tools. We need the implementing technology not just for the profit, but also to build more equitable society, and that inclusive growth of the society it's required, as well as the sustainability of the technology. So without causing any harm to the people and environment, if judiciary honest, blended with novel technologies and supported by appropriate policies, India will dominate in the world of agriculture. This is my last slides. Every culture is bound to fail if agriculture fails. Thank you so much.
Thank you, Bala, for the wonderful exposition.
Thank you, sir. Thank you so much.
Of the cotton story in the country. You moved from BT cotton all the way to organic cotton. It was a very, very big ground that you covered. Thank you very much.
Thank you, sir.
And you have taken almost all of the 1 hour that you had. So that leaves us maybe very little time. I seek your indulgence.
Narayan sir, we have sufficient time. We have 30 minutes for the discussion.
Okay, so there is.
And Bala sir, can I request
you to go for lunch?
Can you please close the screen?
Yeah, I think can we close the presentation, please?
Yes, sir.
In fact, it is so gratifying to see more than 120 participants. Now it has reduced to 115. That means some people are still. Can we close the presentation, Rahul?
Yeah.
Thank you. You can put all the people on the screen, and I already see some questions in the chat box, and one hand is also up. But I also noticed that my good farmer friend, Mr. Ravichandran, is in the group of people who have logged in. Ravichandran, are you still there? Okay. Anyway, Bala, that was wonderful.
I'm very much in the farm, sir. I'm in my farm and listening to it.
Okay, I want to actually give the first chance to the farmer to ask a question. You have recently raised an issue about the cotton pricing, the procurement cost, and
That's
the
right, sir.
difficulty is posing to farmers. Of course, technology fatigue is a major problem, new developments, and I think the BT cotton story in the country is a typical example of how the choking of technology flow can actually take us backward. Tackling new problems, confronting old problems in a different way, and about illegal HT cotton, about which you actually told in an elaborate way. But all these things are also dependent on the market and policies. What was your grouse about the pricing, very quickly?
Yeah. So actually, so because of this,
if you can respond, it will be useful. Okay.
So because of the US tariff, so the price, we haven't even reached that minimum support price of 23.24. I sold my cotton at the minimum support price that that was applicable for 23.24. And again, so when
Ravi sir, we lost your voice. Can you please switch off your camera? Ravi sir?
You switch off your video, sir. I thought the problem was at my end.
No. Ravi sir?
I think, Ravi, you need to switch off your video.
Okay.
Your bandwidth was limiting. Can you repeat the question quickly, very briefly?
See, actually, you were telling about this, pricing. Because of the tariff, US has imposed a 50% tariff. That is one punishment. And another punishment is instead of imposing import duty. And
okay, I think problem which cannot be sorted out within this forum.
I understand.
Discussing the technology.
But
it is good that the technologists are also aware of these kind of issues which can actually influence the dissemination and acceptance of technology. Let us move forward. Thank you for your intervention. I saw a question in the chat box. So, Bala, what is the research that is going on to improve the fiber quality of cotton? That was a question I found on the chat box.
Fiber quality, that is the normally what we do we keep the standard varieties, BT varieties, hybrids, especially RCH2 for fiber quality above 27, 28 only. Whatever the new hybrids or varieties that they are developing, it should be better than the national check or local check. In case of
no, that is fine. That is the standard that you have fixed. But are there any genes identified for fiber quality, for fiber length, text, and all those things? Are there any breeding programs or biotechnology programs going in this aspect? I think that's the update that the seeker wants.
There won't be any. There is no any transgenic varieties or hybrids for improving the fiber traits. Although there are some, we also started one project, so identified some of the genes associated with the fiber, but transgenic there is no any all are in the laboratory condition only, but not yet come to the biosafety level.
Okay. Now there is a question by Suraj Kulkarni. He asks, is the herbicide tolerant cotton really beneficial to the farmers, or is it another marketing gimmick of companies?
Which one, sir?
Suraj Kulkarni's question. I am repeating the question. He has put it on the chat box.
Okay,
so
okay.
You can address it. Is the herbicide tolerant cotton, HTPS cotton, high density planting system, not herbicide tolerance, high density planting system cotton really beneficial to farmers? Or is it another marketing gimmick?
Yes, sir. Sir, HTPS is now it's popular now in Maharashtra and Telangana. So farmers were now we are not able to give the seeds. So many people got impressed. So 30 to 40% of the increase of the yield we reported. Even seeing is believing. Farmers are coming to the field and they are telling that yes, we also want to adopt this high density planting system. Really, this is going to help in the farmers' income, sir. Because this yield is many farmers are. We had conduct that regular meeting. Farmers we are bringing and they are addressing that. How much benefit they got when compared to the traditional methods. So really, this is going to increase the yield of BT cotton.
Okay, so I hope that satisfies Mr. Kulkarni. So it is not a gimmick. There is some real effect. I see that Umesh Kulkarni has raised his hand.
Yeah.
Is he there?
So first of all, thanks. Yeah. Can you hear me?
Yeah.
Umesh Kulkarni.
Yes, sir. Yes, sir.
Yeah. So first of all, thanks, Dr. Bala.
Yes.
Fantastic presentation.
Can you come on the video, please, so that we can see you?
Yeah, because of my network is weak, that's why I put only the audio.
Okay.
And I am repeating the question.
Yeah.
I have two questions, Dr. Bala. Before that, first of all, thanks. It is a very elaborate presentation to understand the status of transgenic cotton status as of now. So first of all, thanks and much appreciated. I have two questions. One question related to the transgenic cotton and one is general question.
Yeah.
So you mentioned that from 2000 to 2004, 2021 to 2024, more or less around 225 BT cotton varieties has been launched, and one of the key challenge is the non-compliance of refuge. And this non-compliance of refuge is we are experiencing since last 20 years, since launch in 2002. So what are the mechanism? Because without refuge, then this technology, whether by ICAR or other companies, is not going to be a sustainable model in the India. So
yes.
How we are going to address this issue? Number one. Number two, non-related to the today's discussion, but related to cotton, is you saw this year is a more unprecedented monsoon across cotton areas, right? Whether from Maharashtra to Andhra, Telangana to Karnataka, everywhere. So with this heavy rain, what are the agronomic tips as a scientific community we are passing to the farmer, because already we are in September end and next 3 months are very crucial to the farmer.
Good, Umesh. I think
we
have got the point of your question. Umesh, I think in the interest of time, you have to make it brief. I think one question was on the weather problems.
Yeah.
The other question was on how do you manage the refugia?
Yeah, correct. So first one is the refugian concept. So this concept has to be followed Because of that, initially the separate packet they were giving, supplying by the BT cotton seed. So farmer used to throw that one. But to avoid that only, now the refugia concept has come. So we'll have to mix 25 gram of non-BT seeds in the BT packet itself, so that the BT non-BT plants are available in the field, so that some of the susceptible population we can maintain it to overcome the resistant development by the insects. Because it's very difficult to completely eradicate any insects, because of that, the survival of the fittest. So a lot of pressure if it's coming to the insect means insects has its own mechanism to break this resistant development. So that's why the refugia concept has come. Now government of India is already told that we'll have to follow. Almost all the companies now, you can see that if you go to the market and find that almost all the companies are putting that non-BT seeds, and also you can see the packet. It's a RAB Lika hoga. So there will be that RAB label also. That means there's compliant with the RAB. Only thing is we have to monitor whether really they are mixing 25 gram of non-BT seeds. That is the only question now. So that we are monitoring now. Almost all the seed testing laboratories, they're getting regularly samples from the seed inspectors. They're monitoring whether the really BT is there or non-BT is there, how much non-BT is there. So this has to be followed. So they are doing already. Regarding the rain, that is not in our hand. So probably
the second part is on the weather problem.
Yes, sir.
Weather, how do you manage the weather vagaries?
Weather, it's very difficult to tell that it's not sporadic rainfall as well as the untimely rainfall. It is going to affect that cotton farmers, especially Maharashtra. Now this also, now of course bole busting is yet to come, but we are expecting a rain during that period also, probably during Diwali period. If you are getting that bole busting, that time rain comes, then it will affect the cotton farmers. It is going
my
to
friend,
be a challenge.
Thanks, Bala. I think my friend Sharad Deshpande has asked an interesting question. We consume cotton oil extensively, but how was it got classified as a non-food crop?
Yes, it's interesting question. Actually, I forgot to cover that one. So one slide I would have put that one. So the Kirti Rathore from Texas A&M University, I was there in his lab in 2001. So that time I saw that there were work going on in the gossip hall. Of course, the government of India is also in talk with them to take that varieties and go for our conventional breeding method to bring that gossip hall free only in seeds, not in other pots. So this is the tissue specific expression, silencing of that particular gene in the only seed, so that we can avoid the seed oil. So that is successful technology. Only thing is now it has the patent. There is some issues. So that has to be imported through proper MOU, so that we can have this technology. This will going to help the industry, especially the oil. We can go for consuming the cotton oil. Of course, already we are consuming 5% of the cotton oil. So once that gossip free cotton is come to India, that we are going to consume cotton oil. That oil import we can curtail it.
Okay, so probably in due course, when the gossip free cotton seed comes, it could be classified as a food crop. Let me move on. I see that Akshay Sable has raised his hand. Akshay, are you there in the group? Because I can't see all of you. Do you want to unmute yourself and come on the video and ask the question? Okay, if he's not there, Ravichandran has raised his hand again. You want to come in, Ravichandran?
Yeah, briefly about this, cotton being affected by rain. This year, in my part, we normally grow cotton in Tanjavur district, summer cotton. This year we had monsoon-like rainfall in February and March. See, in the rice crop, we have this submergence-tolerant rice variety, CR1009, Swarna Sabban, using molecular markers technology. So we address the abiotic stress factors. So like that, is it possible to have submergence tolerant cotton variety using some other technique other than GM, maybe marker resistant technology. That is one thing. And another one regarding this refugia, in the initial years, the refugia seeds were some of the companies provided the tuber as refugia. So pink bollworm being the exclusive pest for cotton, tuber is not, the pink bollworm doesn't affect this tuber. That means that's one of the reasons why pink bollworm developed resistance. Fortunately, in my area, last year we installed about 2,000 pheromones around my village. Not even a single pink bollworm moth was trapped, sir.
Okay, interesting. Regarding your first question, sir.
Yeah, thanks, Ravi. So, Bala, you can ask the question.
Yeah, regarding his first questions, so there is waterlogging is another issue. Waterlogging is another issue, but cotton is not loving the water. It's only heat-loving crop. So cotton water stagnation is going to affect a major issue. But there is no any Transgenic program. But there is a report that there is one kind of glance this cotton is adapting to overcome this waterlogging. But that is not going to help in the completely increase the yield. So waterlogging will be a major issue in some areas. So need a lot of work on all this work on the initial stage only. Waterlogging resistant Transgenic cotton and all under the trial only. We have the program under this our institute. Regarding that,
what about pheromones? They don't seem to trap pink bollworm. So you said that you are using AI to make pheromone combinations. So can you explain that program?
Yeah, now our substation Coimbatore, Dr. Ramesh has developed AI-based pheromone trap, so that we can monitor. So the insect coming, going, everything we can be monitor. It is you will get all the information in your mobile. That program we already launched in Punjab and northern states. We already launched that one. So it's coming in the other state also, because he has developed the AI-based monitoring the insect incidents, especially controlling the pink bollworms.
But Ravichandran's question was that, or his concern was that his traps were not capturing any of the pink bollworm. Is there any?
Maybe that pheromone incident, and also during rainy season, it's very difficult to get that trap.
Okay. Maybe Ravichandran, your anecdotal observation.
Because during rainfall, it won't work, sir. It won't work in the during rainy period.
A problem with the pheromone or something like that. So rather than branding,
so it was during summer, sir. It was during summer. During summer.
Summer means which brand you use, that is also important, because there is the pheromone supplied by whom, that we'll have to see that, because there are many things effective. It won't be effective. That means it's very difficult to catch it.
Yeah, so not even a single rosette was identified.
Okay. So probably who supplied that, we'll have to ask him. So because there will be a lot of issues with the pheromone, the quantity, how much they are in giving, because maximum 40 days it comes. The pheromone, we can keep it 40 days to trap. But I don't know if even single also, if you are single insect also, we are not able to catch, means a problem with the pheromone one.
Yeah, and how about this confused male moth, pheromone disturbance, that is instead of trap, confusing male moth using that pheromone?
AI also, we developed that technique, AI based pheromone trap. So here, this is only to monitor in your mobile from the field. So field, normally we keep the pheromone trap. So the insect coming and going, all those things we can monitor in the AI-based the trap. But the pheromone will be used there also. Pheromone only we are using it. Only this monitoring we are using the mobile technique. That technique is developed by CICR Coimbatore. We are demonstrating.
Thank you, Dr. Bala. I think I see a few questions on the fear of GMOs and the activism and stuff like that, but I would not take it up here. We will leave it
Yes.
for another discussion separately. But I have one last question.
Yes, sir. Yes, sir.
You talked about the public efforts, BNBT and all that.
Yeah.
I remember that many years ago,
Yeah.
NBRI had that Kaivan EC1.
Yeah.
You know anything about the status of that? Because I'm not very clear about what's happening there. Is that program also shelved like the BNBT, or is it
No, sir. That
continuing
was
or pursued?
discontinued. In fact, 2009 it was initiated. We both got 2008, I think, sir. No, sir. Your event also in 2008 we released. The same time, what happened when your order came to us? So we both got the same time only. So what happened? Dr. Tranti was that time he was director. He said that there is a kind of contaminations because he could get that some positive band in the BNBT. So then he reported to ICF that there is some problem with the event circuit. They told. So accordingly,
BNBT is fine. I'm talking about the NBRI. They had a Kaivan EC.
okay.
Dr. Rakesh truly started that work. So
yeah, correct, sir. So that
BNBT,
was also
I
not successful in commercialization. So although we got that one, but we are not able to. We didn't get the positive results.
Okay.
White flag control,
Okay.
CLC, we got that even. So we couldn't get positive control. So that's why we discontinued.
Okay, so it's 24 minutes past 12 o'clock, and I think I should probably step down and then give the floor to the organizers to make some last comments. Thank you all. Thank you very much for a wonderful presentation, and thank you all for a wonderful participation. I know that several people still have some pressing questions. I'm sure Aparna and her team will be sharing Bala's contact details, and I'm sure he will be happy to address those questions. All of them may not have ready answers, but certainly this is a discussion that should continue. Thank you very much, and back to you, Aparna.
Thank you, sir. Thank you so much.
Thank you, sir. Thank you very much. Thank you, Bala sir, for a wonderful presentation. And the list of questions are still coming. The hands are still raised. So I'm, very happy to know that your presentation was really enlightened to everyone and provoked the discussion. But with the respect of time, we have to close the session today. And I will share your email, if you don't mind, to everybody, and they will contact you for their further clarification.
Yes.
And thank you everybody for joining this session. And I'm sorry we are not able to take the question, but definitely we may invite Bala sir once again for the discussion next time, and then we will have much time for much elaborative discussion. So thank you, Narayanan sir, for giving your time, for doing the wonderful moderation of the session. And looking forward to see you again next month with another exciting lecture. Thank you, everyone. Have a nice weekend and a wonderful day to all.
Thank you, madam.
Thank you all very much.
Thank you, madam.
You have a very good day.
Thank you, ma'am.
Thank you. Thank you all.
Thank you. Thank you. Thank you.